And molecular clock is a very powerful idea in evolutionary biology. It basically describes what types of differences you'd expect to see between two different species. So if you compare the DNA sequences from, say, a human and a dog or a chimp and a cat, whatever two species you decide, what types of changes would we expect to see? What rate do they occur? And the molecular clock is an argument that the rate of substitution should be constant. Note that I use the word substitution and not mutation, because when you compare sequences from two different species, say a human and a chimp, any differences that are fixed between them, that is to say that all the humans have one nucleotide at a particular position of the gene and all the chimps have another nucleotide at that position, those are called fixed differences between the species. Those are substitutions. It must have been the case that sometime in the history between, say, the chimp and the human, that you had a mutation occur in one of these two lines. So if you see a difference, a fixed difference in the gene between the two species, it must have been the case that a mutation occurred at some point in time in the history between the two species. This is when mutation occurred. That was later fixed by natural selection, genetic drift, whatever. Okay? So between the time that, so you imagine that between the time the mutation occurred and the time it was fixed, that is to say this mutation went to a frequency of 1, during this time the allele is said to be polymorphic. Or the population, rather, is said to be polymorphic. There's two different alleles in the population during that time, at that locus. What is a substitution? Well, substitution is the combined mutation and fixation. So this is a substitution of these fixed differences between species. And the reason why we use the word substitution and not mutation is because they're combined the mutation process and the fixation process. Okay? Now, for neutral mutations, this molecular clock works for neutral mutations. That is to say, mutations that have no deleterious or beneficial effect on the individuals bearing the mutations. You can actually calculate what's called the rate of substitution, which is usually called K. So that's the rate of substitution. And this is a population genetics result that's pretty easy to derive. So it only involves one additional parameter over which you guys that you have not seen. We call it U. This is the neutral mutation rate, or neutral mutation probability might be a better word for it. This is the probability that you have a neutral mutation occurring at some locus. Now, what is the rate at which these mutations are introduced into a population? This is to say this is the probability that one chromosome in a population changes. How many chromosomes do you have in a population? You have 2 N chromosomes in a population. So the total number of neutral mutations that are introduced every generation to a population is 2 times the population size times the neutral mutation rate. Is that clear? So this is the probability that any chromosome has a neutral mutation occur in a particular generation. This is the total number of chromosomes in the population. So this is the total number of neutral mutations that are introduced every generation to a population. What's the probability that any one of these is fixed? Well, I told you what that is, the probabilities. I told you that the probability that any allele when it first occurs, any neutral allele when it first occurs becoming fixed is 1 over 2n. So one of the interesting results in population genetics is that the substitution rate is equal to the mutation rate, neutral mutation rate. Right, so when you actually compare different species, I mean, if you don't see it, it's called canceling, right? The 2n's cancel, right? So that's a pretty simple result to get out. But this just describes the dynamics, what types of changes, the rate at which changes between species accumulate for neutral mutations. Okay, so that's the last thing I wanted to talk about. The molecular clock, I mean, this is an idea that if you become an evolutionary biologist or you take the upper division of evolution class here, you'll hear about molecular clock a lot more. So this might be the first time you've heard it. And there's the math. Okay, the last thing I want to talk about is gene flow. So gene flow involves, remember I told you about one of the models we have in population genetics is even the concept of a population, right? And I told you that every species is composed of at least one population, where populations can be thought of, at least in this idealized world, as individuals within populations can randomly mate one with the other. But individuals from this population have a lower probability or lower chance of mating with individuals from this population. Okay? So gene flow is just the following. You can also call it migration. It's migration of individuals from one population to another. So for instance, for an individual from this population to mate with an individual from that population, first of all, it has to migrate to that population, then it has to mate with that individual, right? So that's why the probability of an individual from this population mating with an individual from that population is lower. You have to first migrate over there. But, and we will talk more about gene flow when we talk about speciation, right? Some of the simple models of speciation, how different species are formed in the future lecture. But the idea here is that how can gene flow change allele frequencies? Well, if an individual from this population moves over here and has a particular allele that's either rare, it'll change the frequencies of alleles over here. Right? So for instance, maybe this individual has a new mutation. The mutation occurred over here. The only way for this mutation to get over into this population is for it to either independently occur over here, which might have a low probability of occurring, or for an individual that bears that mutation here to move over to here. Right? So this is just gene flow. So there's gene flow is of vast importance in terms of understanding how speciation occurred, but it also is of some practical importance. And so one of the concerns that people have with genetically engineered crops, for instance, can boil down to gene flow. That is to say, as an example, I have in the notes an example involving rice, a particular strain of rice called Liberty rice. Make big. There was this particular strain of rice was genetically engineered to be resistant to a herbicide, which is great for farmers. It's unambiguously good for farmers, right? Because if you can spray a herbicide over your rice, then you knock out the weeds but leave the rice, and the rice will have less competition and you'll get more rice. So that's good, right? Or at least that was the idea. But it was only grown in a couple of test plots, right? They modified it. It was never actually approved for human consumption, but it was grown in a few test plots. But the problem is the gene that they inserted into the strain that caused the herbicide resistance got into the rice that is actually grown for consumption. Right? This occurred around 2000 is when this occurred. And it caused a lot of problems in terms of the for rice growers. First of all, the cost of rice dropped dramatically because people didn't want to buy the rice that was potentially had this herbicide in it, herbicide-resistant gene in it. And even Japan banned the importation of rice from the United States for a while because of this. Right? Japan's a major importer of our rice. Right? So here's an example of gene flow that has real consequence, where you imagine you have the Liberty rice over here in this test plot, and through gene flow it got into the rice for consumption. That is an example of gene flow that has some practical importance. Okay, what is the time? Oh yeah. Okay, so I must just be getting slower as I get older. All right, so now so that's all I want to do to talk about for population genetics. Okay, so what I'm going to be doing now is starting my lecture on examples of natural selection in the laboratories and in the wild. Okay? And I think I will play a slideshow. Unblank the screen. There we go. So the first thing I'm going to talk about is experimental evolution. Running out of board. Okay, so experimental evolution. So this is evolution that's performed under controlled conditions in, say, laboratories. And it's been done for a long time. Almost as soon as people realized that they could artificially select in a controlled way, and they did. Of course, we already know about domesticated animals and plants, but that was sort of more seat-of-your-pants type of selection. There's an example of a very long-term selection experiment that goes back to the 1890s. It's in Indiana, and it's an experiment on corn. Okay, and specifically, it's an experiment where they've selected for high and low variants of strains of corn that have very high oil content and very low oil content. So this is, if you wonder about corn, the wild relative of corn is teosinte, and if you look at teosinte, it doesn't look much like corn at all, but through artificial selection and using sort of seat-of-your-pants type of selection, people normally practiced it for a long time, they're able to get corn that looks like the stuff you see in the supermarket today, lots and lots of kernels. What these people in Indiana did is they selected, this is what the initial oil content was in the corn in the 1890s. Over time, every year, they would select for, in one test plot, they would take the corn that had the individual plants that had corn that had the highest oil content, and then they also had another test plot. They said, okay, let's do the opposite. Let's select for individuals or corn individuals that have low oil content in the kernels. Okay? And you can see that they got this dramatic increase or decrease in oil content in the corn in the kernels. You might ask what happened over here. This is sometime about around either right before or after World War II. They decided to take the high content ones and make two more plots. Continue to select for high oil content, but also then had another plot where they actually started to select for low oil content. And then again here, they did a split at the same time in the low oil content. Let's continue to select for low oil content, but let's split it and see if we can get high oil content back. The point here is this is just selection. You humans choosing which individuals to allow to reproduce, and you can get dramatic increases or decreases in the oil content in this particular example. Right? Now the idea here is that there's variation among individuals that has a genetic basis for oil content in the kernels. If there were no genetic variation, you could do all the selection you want, but you get no response to selection. Okay? So here's another example. I'll do this example next. So these are two people I know. Holly Whitman is a professor of evolutionary biology at the University of Idaho, and Jim Bull is a professor at the University of Texas. He was actually co-advised by Jim. He's an interesting fellow. When we talk about sex determining mechanisms, he's the guy who discovered that turtles and crocodiles have temperature-dependent sex determination. So he was a herpetologist originally, but then he changed his interests and started studying viruses. And the viruses he initially studied, and then Holly became interested later on with them, are what they call bacteriophage. I'll write that down. Sometimes people just call them phage, but these are viruses that infect bacteria. That's all they are. They're viruses. Here's an example of the virus pictured using a scanning electron microscope. Here's a schematic of the genome. So the particular virus they studied was Phi-X174. So it's a crazy name, but viruses have crazy names, right? So this particular virus is called Phi-X174. It's a double-stranded DNA phage, bacteriophage. Its genome, is circular, right? It's a big circle, and the genome size is 5,382 nucleotides, if I remember that correctly, about 5,400 nucleotides. It turns out that Phi-X174 was the very first thing that was ever sequenced that had a full genome sequence. This occurred in the late 1970s. If you think about the human genome was sequenced in 2000, this is the very first genome of anything that was sequenced with Phi-X174 in the late 70s. And I think it has 13 genes. Here's where the different genes are. There's an F gene, very imaginatively named, J gene, the E gene, the D gene. It's kind of crazy because there's overlapping reading frames in this virus. That's not too exciting, but that's just to say there's 13 genes. We know where they are. That they're easy to sequence. So what do? Well, in their two laboratories, independently, I should say, so these infect bacteria, but not just any bacteria. They infect E. coli. E. coli is the bacterium in your gut. E. coli grows really well at 37 degrees Celsius, which also happens to be 96.8 degrees Fahrenheit in your body temperature. It's well adapted. E. coli is well adapted to grow at 37 degrees Celsius. These viruses are also well adapted to infect E. coli at 37 degrees Celsius because that's the temperature in which they work best, or they've evolved to work best. So what did Holly and Jim do? What they did is they said, well, let's change the temperature in which we grow the E. coli. Right? So the idea being they have actually, the crazy thing is Jim would make all these crazy things in the lab. He really liked tinkering. So he had what he called what's called a chemostat. But he had basically a flask, or a tube really, and he had that tube in a water bath, which he could control the temperature. He could make it 37 degrees, he could make it hotter. Then he had a broth, a nutrient that he kept pumping in, and then he also had LB with E. coli. So he had a growing culture of E. coli over here, and he dripped the E. coli in, and then he sucked some of the E. coli out, so the volume in this tube would be constant. So in this tube here, you have the E. coli, and you have the Phi-X174. So what happens? Every time a little E. coli drops into this flask, there's a bunch of Phi-X174 waiting around, and they latch on, they get in, and once they get into the bacterium, they replicate. And once you have maybe a couple hundred viruses inside that bacterium, they explode the bacterium, and they go out in the environment looking for more E. coli. So it's bad for the E. coli, right? They exploded and have lots of little viruses come out. So he kind of likened this to dropping babies into a pit with crocodiles. That's kind of how he described it. It's kind of gruesome. That's Jim for you, right? So anyways, that's the situation. So the point being that you're always dropping fresh, naive baby E. coli into this flask, and the viruses are continually replicating, and in this case, evolving. Now what they did is they took the water bath, which normally you'd have at 37 degrees Celsius, and they changed the temperature to 43 degrees Celsius. What happens? Well, initially the viruses are not happy. They do not grow well at 43 degrees Celsius. Now let's go back to the slide, please. But that doesn't last for long. So what this is 0 is the fitness at 37 degrees. You change the temperature to 43 degrees, and the viruses do not grow as well. In this case, growth is a proxy for fitness, right, in terms of the population genetic sense. But look what happens after a few days. In the Texas experiment, after about 4 days, you see that the viruses are doing really well. They're growing quite well, right? So their rate of growth increases over time, and you see the same thing occurring in the Idaho ID experiment, right? Same thing. The same experiment, same organism, two different places, two different states even. So this is an example of evolution in the lab. The response to growth at 43 degrees has changed over time. In this case, changed over just 10 days. Remember, you can get very rapid evolution in viruses because their generation time is so rapid, in the order of 40 minutes in this case, and the population sizes are very large, and the mutation rates are very high. So what they did after the experiment is they went into the experiment, into the flask, and they asked, okay, for the viruses at different time points, at different days in the experiment, what were the mutations that were in the genome? Remember, they could sequence the entire genome. They knew all the variants in the genome that were around in the population. And this is the mutations that they saw. The order in which they appeared, the position. Remember, this is the nucleotide position from 1 to 5382, the total number of nucleotides in the genome. So this is the position of the genome. And this is what type of mutation it was. So this is basically a replacement change from the amino acid that has a Q to R. I always forget what the amino acid symbols are. Right? But they're two different amino acids. So you can see, first of all, that almost all of the almost all the mutations were mutations that changed the amino acid that was coded for. And that's not necessarily what always happens. So remember, when you have a mutation in a protein coding gene, first of all, you guys all remember that you have codons, right? So gene, you have triplets of nucleotides that code for an amino acid, right? There's 20 amino acids, but if you have a triplet of nucleotide coding for an amino acid, you have 4 to the third possible nucleotides, right? Which is 64. So there's 64 possible codons that code for 20 amino acids. So the point here is that there's redundancy in the genetic code. That you have up to 6 different codons coding for the same amino acid. So when you have a mutation in a gene, it can be one of 2 sorts. It can be a replacement change. Sometimes that's more frequently called a non-synonymous change. That is a mutation that changes the amino acid that's coded for. The other alternative is that mutation would be called synonymous. You have a mutation in the codon, or in the DNA, but it doesn't change the amino acid because of the redundancy in the genetic code. Okay, so you have non-synonymous and synonymous changes. The interesting thing is that you have a real excess of non-synonymous changes occurring in this experiment, which is also some evidence that they're adaptive in nature. Also remember, you can actually measure the fitness. You can actually measure the effect that each one of these mutations has on the rate of growth of the viruses. And one of the interesting things that happened in this experiment is, so there's 2 interesting things to point out about this experiment. First of all, you can actually see the mutations sweep through the population. This is the day of the experiment and their estimates of the frequencies of these different mutations at different times. So the first thing is, this is really, you can go into the population and really see the mutations, if not occurring, changing in frequency very rapidly. And just as an aside, in this experiment, they often saw the same mutations being responsible for adaptation to high temperature in both experiments. That's probably more something that's peculiar to this system and not something that's general, but they also saw repeated evolution that is to say the same mutations being responsible for adaptation. That's it for this time.